Hello everyone! Welcome to this presentation on HPLC method validation for quantification of tetrahydrocurcumin in bulk drug and formulation by Raut and Shaji, published in 2021. In this video, we will be discussing the objective of the study, the procedure used, the parameters for method validation, results and discussion, as well as the conclusions and recommendations.
Tetrahydrocurcumin is a potent antioxidant and anti-inflammatory agent found in turmeric. It has been found to have various pharmacological activities, including anti-cancer, anti-diabetic, and neuroprotective properties. Therefore, it is essential to develop a reliable analytical method for the quantification of tetrahydrocurcumin in bulk drug and formulation to ensure the quality and efficacy of the product. By developing a validated HPLC method for the quantification of THC, the study aimed to provide a valuable tool for the quality control analysis of THC in bulk drug and formulation. The main objective of this study was to develop a reliable and accurate method for the quantification of THC in bulk drug and formulation using high-performance liquid chromatography analysis. The researchers aimed to validate the method by determining its specificity, linearity, accuracy, precision, and robustness. The method was validated using the International Conference on Harmonization Guidelines for Method Validation.
The researchers used HPLC for the analysis of THC in bulk drug and formulation. The HPLC system comprised of a JASCO PU2089 HPLC pump equipped with JASCO 2070 UV vis detector. The schematic diagram of the procedure is as follows.
For the preparation of standard solution, 10 mg of THC was accurately weighed and transferred to a 50 ml volumetric flask. 35 ml of methanol was added, and with the aid of sonication, it was dissolved completely. Volume was made up and the solution was mixed well. 10 ml of aliquot was withdrawn and diluted to 50 ml with diluent. For the preparation of sample solution, 500 mg of the 1% weight per weight THC gel was weighed on a butter paper and transferred the gel along with the butter paper to a 50 ml beaker containing 25 ml of diluent. The sample was sonicated for 5 minutes to form clear solution, and the contents of the beaker were transferred to a 50 ml volumetric flask. The beaker was rinsed 2 times by 10 ml of methanol and transferred the same to the volumetric flask. The volume was made up of the mark with diluent and mixed well. The solution was filtered through a 0.45 micrometer nylon syringe filter, discarding 3 to 5 ml of sample. 10 ml aliquot was further diluted with methanol to 25 ml. For system suitability, filter study is carried out to ensure that no drug is retained by the filter during sample preparation. The solution was collected after passing through a 0.45 micrometer PVDF filter and a 0.5 micrometer nylon filter by discarding 5 ml solution. The results were compared to unfiltered centrifuge sample solution. For specificity, the mobile phase or the excipients of the formulation should not interfere with the analysis. The developed method needs to be specific. The placebo solution, THC solution, and gel solution were injected and peak purity was determined. For linearity, THC solutions at 5 levels were prepared from 10 to 150% of working concentration. At each level, analysis was carried out in triplicate. The peak areas versus concentrations data was evaluated by linear regression analysis. 3 datasets corresponding to triplicate analysis were constructed. These data was analyzed by the test one-way ANOVA with post-test Turkey using 95% of confidence interval. An accuracy study was performed by adding known amounts of THC to the placebo preparation. The actual and measured concentrations were compared and recovery was calculated. Recovery of the method was evaluated at 3 different concentration levels, corresponding to 50, 100, and 150% of test preparation concentration. For each concentration level, 3 sets were prepared and measured. The precision of the assay method was evaluated in terms of repeatability by performing 6 independent assays of THC. Under the same experimental conditions, intermediate precision of the method was checked by another person performing the same procedure on a different day. Percent RSD not more than 2 was taken as the limit. For the robustness, the factors chosen for this study were the change in wavelength, flow rate, and column temperature. The appropriate amount of THC, bulk drug, and formulation were weighed and diluted with methanol. The effect of change parameters of analysis of THC in bulk drug was evaluated in terms of RT, asymmetry factor, theoretical plates, and assay.
The HPLC method was validated for the following parameters. System suitability. The system suitability testing checks if the chromatographic system is suitable for analysis. The method was used to analyze 6 THC samples. Results are then compared to unfiltered centrifuge sample. Specificity. The specificity of the method was determined by analyzing the standard solution and sample solution in the presence and absence of interfering substances. Linearity. The linearity of the method was evaluated by analyzing the standard solution of tetrahydrocurcumin at different concentrations. Accuracy. The accuracy of the method was evaluated by analyzing the recovery of tetrahydrocurcumin at 3 different concentration levels. 4. Precision. The precision of the method was evaluated by analyzing the intraday and interday precision. And last, robustness. The robustness of the method was evaluated by making slight variations in the chromatographic conditions.
The results showed that the system's suitability parameters, including retention time, peak area, and tailing factor, were within the acceptable range, indicating that the HPLC system is suitable for the analysis of THC. Therefore, the method can be considered reliable and suitable for routine analysis. The authors demonstrated the specificity of the method by showing that the THC peak is well resolved from other impurities in the sample. The peak purity of THC was found to be greater than 0.99, indicating the absence of impurities. And also, the method is selective for THC. The linearity of the method was determined by analyzing THC at different concentrations ranging from 10 to 100 micrograms per milliliters. The calibration curve was found to be linear with correlation coefficient of 0.999, indicates that the method can accurately measure the concentration of THC over a range of concentrations. The accuracy of the method was evaluated by spiking known amounts of THC into a placebo sample at 3 different concentration levels. The percent recoveries were found to be in the range of 98.5 to 101.5%, indicating good accuracy. The precision of the method was evaluated by analyzing 6 replicates of THC at 3 different concentration levels. The percent relative standard deviation for intraday and interday precision was found to be less than 2%, indicating good precision. Both results of accuracy and precision indicates that the method can produce reliable and reproducible results. The robustness of the method was evaluated by making small deliberate changes in the chromatographic conditions. The method was found to be robust because the changes did not significantly affect the results, indicating that it can produce reliable results even with minor changes in the method parameters.
HPLC method validation was studied as an analytical technique to be used in determination and quantification of tetrahydrocurcumin in bulk drug and formulation. This method was investigated for its system suitability, specificity, linearity, accuracy, precision, and robustness as parameters in the method validation. The results showed that the new HPLC method indicated an outstanding system suitability, specificity, and linearity, having a significantly low percent error in the stated parameters. Furthermore, the method also displayed exceptional accuracy, precision, and robustness, as observed in the results. As all of the above points have demonstrated, this method is notably suitable and optimal for quantification and routine quality control analysis of THC, which is highly beneficial in the development of the medical and pharmaceutical research and manufacturing industries. Further studies can be conducted to investigate the applicability of the method for the analysis of tetrahydrocurcumin in biological fluids and other formulations. Thank you for watching this presentation. If you have any questions or comments, please feel free to share them in the comment section below.